So in this section, we'll address the nerve-muscle relationship. Skeletal muscle never contracts unless it's stimulated by a nerve or artificially with an electrode. If the nerve connections are severed or poisoned, a muscle is going to become paralyzed. And if the connection is not restored, the paralyzed muscle undergoes a shrinkage called denervation atrophy. Thus, muscle contraction cannot be understood without first understanding the relationship between nerves and muscle cells. We have somatic motor neurons. These are the nerve cells whose bodies are in the brainstem and the spinal cord that serve the skeletal muscles. The axons of somatic motor neurons are called somatic motor fibers, and they lead to the skeletal muscle. Each nerve fiber branches out to several different muscle fibers, and each muscle fiber is only supplied by one motor neuron. So one motor neuron could supply several muscle fibers. However, each muscle fiber only has input from one motor neuron. That brings us to discussion of a motor unit. A motor unit is a nerve fiber and all the muscle fibers it innervates. Here we're showing two different motor neurons, a red one and a blue one, and they branch towards the end and innervate different muscle fibers. You can see that these muscle fibers may be mixed up amongst each other and dispersed throughout the muscle. The muscle fibers that are innervated by one motor neuron will all contract in unison and produce a weak contraction over a wide area. This provides the ability to sustain long-term contraction as motor units take turns contracting. So the blue ones might be in contraction while they're relaxing, the red ones are in contraction. This allows sustained contraction of muscle that will help us in our general postural control. An effective contraction usually requires the contraction of several motor units at once. The way that we get more strength in contraction is by recruitment of more motor units. The average motor unit will encompass 200 muscle fibers. Small motor units are for fine degree control, and larger motor units provide more strength and control. In a small motor unit, we may only see 3 or 6 muscle fibers per neuron. In the hand muscles, for example, we need a very fine degree of control, and the muscles around the eye, we also need a fine degree of control. In some of the largest motor units, like that operates the gastrocnemius, there are 1,000 muscle fibers per neuron. These contractions are very powerful and thus need to be supplied by large motor units. They don't require a lot of fine control. The point where a nerve fiber meets its target cell is called a synapse. When the target cell is a muscle fiber, that's called a neuromuscular junction or motor end plate. Each terminal branch of the nerve fiber within the neuromuscular junction forms a separate synapse with that muscle fiber. At the point where the neurons connect with the muscle cell, there'll be a small indentation, sort of like a handprint that's pressed into soft clay. The individual synapses would be like the points of where your fingers connect to the clay, and then your arm would be like the neuron coming down. Thus, one nerve fiber is synapsing with the muscle fiber at several different points in the neuromuscular junction. In each synapse, the nerve fiber will end in a synaptic knob. This would be the synaptic knob. In the synaptic vesicles of somatic motor neurons will be filled with acetylcholine. This is the neurotransmitter that will be released across the cleft in order to stimulate the muscle fiber on the other side. The synapse has a space between it. There's a space between the synaptic knob and the muscle sarcolemma. This is called the synaptic cleft. And each motor neuron is covered by Schwann cells. We'll learn about this in the section on neurobiology. The Schwann cells wrap themselves around to insulate the neurons and help the signal get carried long distances. These Schwann cells will then isolate the whole neuromuscular junction. When the signal is passed from the synaptic knob to the muscle membrane, the sarcolemma, the synaptic vessels will pass out of the synaptic knob by exocytosis, and thus they release the neurotransmitter into the synaptic cleft, this space right here. On the other side of the synaptic cleft, there are acetylcholine receptors. These proteins are incorporated into the muscle cell plasma membrane. Now, you can sort of ignore this fact, but there are junctional folds in the sarcolemma beneath the synaptic knob. The junctional folds are these indentations. And you can see that the acetylcholine receptors are not just simply straight along the sarcolemma. These indentations in the sarcolemma allow increased surface area and thus more receptors for the acetylcholine. The entire muscle fiber, as well as the Schwann cell, is coated by this collagen and glycoprotein layer called the basal lamina. This separates the Schwann cell and the entire muscle cell from its surrounding tissues. So once we have acetylcholine in the synaptic cleft and it's activating these receptors and eliciting a response in the muscle, how do we stop it? Acetylcholinesterase is the key. This is an enzyme that breaks down the acetylcholine after it's caused a muscle contraction. So it breaks down acetylcholine in the synaptic cleft. It prevents it from binding to these receptors, and it reuptakes the remaining parts. We'll look at that in more detail here in a moment. Here you can see the neuromuscular junctions where we have the neuron motor fibers coming down to meet the muscle. Here's the neuromuscular junction where the synaptic knob is pressing down into the cell membrane. So this coupling of the neuron and the muscle fiber can be a little tough to understand. So I found another animation for you. This one's produced by W.H. Freeman, comes along with their texts. Hopefully this clears it up just a little.
Communication between neurons and communication between neurons and muscle occurs at specialized junctions called synapses. The most common type of synapse is the chemical synapse. Here, we examine the events that take place at the neuromuscular junction, a chemical synapse using the neurotransmitter acetylcholine. Synaptic transmission begins when the action potential reaches the axon terminal. The resulting depolarization, due to the opening of voltage-gated sodium channels, initiates the sequence of events leading to the release of transmitter. Depolarization of the axon terminal membrane causes voltage-gated calcium channels to open, and calcium ions rush into the axon terminal. The calcium ions trigger the release of neurotransmitter by causing the synaptic vesicles closest to the active zone of the synapse to fuse with the presynaptic membrane. This fusion process is regulated by an interaction between protein complexes expressed on the vesicle and presynaptic membranes. When the vesicles fuse with the membrane, they release their content of neurotransmitter into the synaptic cleft. The neurotransmitter moves across the cleft and binds to receptors on the postsynaptic membrane. The physiological response of the postsynaptic cell depends upon the particular neurotransmitter and receptor combination. In this example, acetylcholine binds to an ion channel. The channel opens, and sodium ions enter the postsynaptic cell. The signal is thus propagated to the postsynaptic cell. After a neurotransmitter has achieved its effect, it must be inactivated. For acetylcholine, an enzyme in the synaptic cleft, acetylcholinesterase, breaks down the acetylcholine into acetyl-CoA and choline. The release of transmitter from the receptors caused the channels to close. The choline is then taken back up into the synaptic terminal for resynthesis into acetylcholine. The synaptic vesicles are also recycled via endocytosis of the presynaptic membrane. The recycled vesicles are later refilled with neurotransmitter molecules and are ready for another round of synaptic transmission.
Before we move on to the next section, where we investigate these further processes and the role of sodium and so on and so forth, please take a moment to diagram the neuromuscular junction. You don't have to include the basal lamina, and you don't have to include the junctional folds. You can just make it look straight across like this. This is a perfect neuromuscular junction for the purposes of this class. After you've got your diagram done, we'll move on to the next section where we look at the behavior of skeletal muscle fibers and excitation, contraction, coupling.